Hello everyone, I welcome you all in a series of the lectures in biomolecules, and today is our 9th lecture. Before going to the details of the lecture, I will like to give a recap of my last lecture. In the last class, I was talking about the structure of proteins, and there we discussed about the all kind of structures which has been divided particularly in 4 categories. Primary structures, where we focus on number of amino acid and the disulphide bonds, particularly number and type of amino acids. In the secondary structure, we look over the segment, the repetitive segment, that how they have arranged in the protein, and how are they kind of having structure, what kind of structure they are having. And when we looked in the details, there we found that 2 types of structures are there, alpha helix and beta pleated seed. In the alpha helix, we saw that coil kind of structure is there, and in the beta pleated seeds, we saw that how by hydrogen bonding 2 chains are kind of arranged one above the other. It can be parallel, it can be anti-parallel. Then we talked about tertiary structure. So, in tertiary structure, we were talking about that so tertiary structure, basically it is a 3-dimensional arrangement of all atoms in the protein. It is a 3-dimensional arrangement of all atoms in the protein. Protein folds spontaneously in the solution to maximize their stability. Spontaneously in the solution to maximize their stability. How will it protein folds spontaneously in the solution? Basically, what happens that when it folds, it starts having bonding, hydrogen bonding in between the different segment of the protein, and because of that hydrogen bonding, it gets stable and its energy gets minimizes. So, as long as it's keep on reducing its energy, and we know that it's a nature's law that every entity in the nature wants to be in the minimum energy state, and that's what happening by bonding, it releases energy, and that leads to the stabilization of the protein. So, it in the solution it tries to fold, so that by folding it will have different kind of bonding in a different segment of the protein, and that leads to the maximum stabilization. Okay. Every time there is stabilizing interaction, that between atoms that releases free energy. So, every time there is a stabilizing interaction between atoms, that releases free energy. Okay. And the more free energy is released, the more stable the protein is. So, if you are having kind of linear structure, consider this is linear structure, and if it gets folded, these foldings providing a maximum possibility of bonding in the same basically peptide chain, and that is how it gets stabilized. So, what kind of these stabilizing interactions are? I told that every time there is stabilizing interaction between atoms, that releases free energy, and because of the release of this free energy, it gets stabilized. What are the kinds of these interaction? So, stabilizing interaction. Let us talk about the stabilizing interactions. Stabilizing interaction in protein include disulphide bonds, so consider if the protein is having 2SH group, it can lead by folding, and the 2SH group can get converted to the disulphide bonds. It can get converted. So, this possibility can happen. Okay. This is disulphide bond. Second is hydrogen bonds. First, I am giving this general example that how this stabilizing interaction helps this protein to be stable, and that leads to the tertiary structure. Now, after this I will again give one individual example, and with that I will explain it. Yeah. So, hydrogen bonds. So, as I mentioned that basic as well as acidic part in the same peptide chain can lead to the hydrogen bonding. Basically, if you have carbonyl group as we have seen, and carbonyl oxygen and NH of the amide bond of the two different amide groups can lead to the hydrogen bonding. So, that possibility also exists. Then third is electrostatic attractions. Electrostatic attractions. Electrostatic attraction. And fourth is hydrophobic interaction. Hydrophobic interactions. All right. So, these are the stabilizing interaction between the peptide groups, basically can lead to the tertiary structure. Now, before going to the quaternary structure, I will again like to categorically explain that how primary structure, secondary structure and tertiary structure gets arranged. So, what happens in the, as I mentioned, in the tertiary structure gives a specific overall shape to the protein. Tertiary structure gives a specific overall shape to the protein. What does it mean? As I mentioned that alpha helix, the coiled structure, where we saw that two repetitive units can come, particularly two amino acid which are having 4 residue gap that can get involved in hydrogen bonding. Okay. Another is beta pleated sheet. In the beta pleated sheet, again carbonyl of one peptide group and NH of another peptide group can involve in hydrogen bonding. So, that is the another, and it can be parallel or anti-parallel, two types are possible. So, tertiary structure gives a specific overall shape to the protein, and it involves interaction and cross links. Cross links between different part of the peptide chain, between different part of the peptide chain. Different part of the peptide chain. I have already mentioned that it can be stabilized by, again I am repeating, as I mentioned that there are stabilizing interaction. Again I am repeating just to put emphasis on those interactions. So, it can be stabilized by hydrophobic interactions, hydrophobic and hydrophilic interaction, hydrophobic and hydrophilic interactions. Hydrophobic and hydrophilic. So, what is hydrophobic interaction? Hydrophobic interaction means like alkyl group are there. So, alkyl group, if they basically interact, so these interactions are aryl group are there. So, their interactions are called hydrophobic interactions, whereas hydrophilic interaction. What is hydrophilic interaction? Hydrophilic interaction is basically if there is possibility hydrogen bond formation. So, if substituent is consider CH2OH, and if you have in the vicinity, if you have water molecule, so with that it can give the hydrophilic interaction. So, it has either water molecule or alcohol. So, this interaction is called hydrophilic. This is also stabilizing interaction. Okay. Then second is salt bridges. Salt bridges formation is also stabilizing interaction. Salt bridges. So, what is salt bridges? Salt bridges basically if you have carboxylate group in peptide chain. So, carboxylate group will have negative charge, and in the same peptide chain if the amino group is in the ammonium form basically. So, that will be positively charged. So, to make it more clear, I am putting here negative and here positive. Now, these two, carboxylate is negatively charged and ammonium is positively charged, it will have interaction, ionic interaction among themselves. So, salt bridges. Then the other salt bridges basically it becomes like charged interaction. Then third one is hydrogen bonds. Hydrogen bonds that I have already mentioned that hydrogen bonds are possible. So, carbonyl, and if the hydrogen is available with the alcoholic substituents or amidic substituents, any possibility will help. So, this alcohol one I showed, and then again for the amidic interaction. So, this is another and the final one I mentioned that disulphide bridges, that disulphide bridges this is another stabilizing interaction. So, now I am assuming that I have completed that in the tertiary structure what kind of stabilizing interactions are possible. We started with the hydrophobic and hydrophilic interaction, then we moved to salt bridges. Salt bridges are possible if we have in the polypeptide chain, if we have carboxylate group and the ammonium group. So, there that interaction is possible. Then we came to the hydrogen bond. In a hydrogen bond also we saw that it can interact with the alcoholic or amidic NH, or I can say that amine NH or and finally, I mention about disulphide bridge formation. So, these are the basically stabilizing interaction. To compile all together, I will like to make a structure where these all interactions whatever I have mentioned about the tertiary structure I will like to incorporate. So, let me draw this structure. Please see this drawing carefully, and that will help you in. So, here I am making the helical structure to basically repeat the kind of what to say, we are having alpha helix as I mentioned in the tertiary structure, and then I talked about the beta pleated sheet over here. So, this will again beta pleated sheet and in the segments, and then again I am making here helix, and that will give you. So, to make it better I will just in the tape form I am making this picture, so that it will get more clearer, and so this is the helix part, and this is I am denoting here this is here beta pleated sheet part actually. So, this is a beta pleated sheet part again, and this is rest of the. I am just making this yeah. So, now I completed this, it is a big polypeptide chain. So, now I will make all the structure. So, let me first show you basically salt bridges. The salt bridges, to represent salt bridges I am putting ammonium as well as the ester part. So, so that so this is the salt bridges, and then I will hydrogen bonding, to represent hydrogen bonding in the different segment I will put here CH2 OH and another CH2 OH group. So, this is another hydrogen bond. So, let me write here, this is salt bridge this is salt bridge and this is hydrogen bond. Just I am making my drawing a bit more clear. So, you should not have difficulty in understanding. Okay. Hydrogen bond. Now, I will put the beta pleated sheet. If you consider the two parallel chains are there and there this hydrogen bonding is taking place, I will note it with the this hydrogen bonding. So, this is beta pleated sheet. Beta pleated sheet. Okay. This also has hydrogen bonds. So, hydrogen bonds again here. Now, I will talk about the disulphide bridges. So, disulphide bridges, as I mentioned that if thiol groups are there and that will lead to the disulphide bonds. Disulphide bonds, that is also stabilizing interaction. Now, hydrophobic interaction, let me mention some hydrophobic interaction. For hydrophobic interaction, I am putting here benzyl group and the another benzyl group consider from the amino acid. So, this interaction and basically you can have here even CH3 also. So, these interaction. So, CH3 group. So, this is called hydrophobic interaction. Hydrophobic interaction. Hydrophobic interaction. Hydrophobic interaction. And this is, you can see that as I told you, this one, these all are like tertiary structure. This is alpha helix. Alpha helix, okay, and in alpha helix also we have hydrophilic interaction. So, to demonstrate hydrophilic interaction, I will make here, as I mentioned, that OH will interact. So, consider it has phenolic OH and the other one is having CH2 OH. So, this interaction is hydrophilic interaction. So, hydrophilic interaction, hydrophilic interaction basically to water, because they are having OH group. So, and another I will show here salt bridges again. So, this carboxylate and here you are having regular amide group that can also have this hydrogen bonding. So, I hope here I have tried to demonstrate all kind of stabilizing interaction through this big polypeptide chain, and there I have mentioned all stabilizing interactions. So, now I will like to exemplify basically. If you take an example of globular proteins are spherical in shape, and to exemplify it, I will take the myoglobin. What is the role of myoglobin? Basically, myoglobin transport oxygen into the muscles, basically oxygen to muscles, and where because of that we get energy. And the example of this was the globular protein, and example of fibrous protein, long fiber like shapes is alpha keratin, which makes up hair, skin and nail basically, and feathers contain beta keratin. So, let me write down so that you should not forget. So, globular protein, exemplified by the myoglobin. Myoglobin basically carry out synthesis, transport and metabolism in the cells. These carry out, globular protein carry out synthesis, transport and metabolism. In the cells. And myoglobin basically transports oxygen to the muscles basically. Fibrous protein, now let us example of fibrous proteins. Fiber proteins consist of long fiber like shapes. Fiber like shapes, and for example, easy alpha keratins, which makes up hair, wool, skin and nails. So, fibrous protein consists of long fiber like shapes, and to exemplify it, alpha keratins make up hair, wool, skin and nails. Okay. Feather contain beta keratin. Feathers contain beta keratin. Beta keratins with large amount of beta pleated tertiary structure. As you can see that structure of feather with large amount of beta pleated tertiary structure. Seat structure, beta pleated. Okay. Let me make some drawing so that you can understand well. Let me draw the basically alpha keratin structure. Here I am making representation for alpha keratin, basically to just I am trying to depict alpha helix structure. As you can see that coiled structure is there. So, this one can give you feeling that how these coiled structures are. So, this is the alpha creatine, this alpha helix basically. Okay. Now, I will move to quaternary structure. Basically, I will move to quaternary structure. What is quaternary structure? So, this much about the tertiary structure. Now, let us talk about the quaternary structure. Quaternary structure. In quaternary structure, some proteins have more than one polypeptide chain. Now, in the quaternary structure, a protein, the individual chains are called subunits, and how are they arranged in respect to each other. So, in the quaternary structure, basically it has more than one polypeptide chain, and these polypeptide chains are how arranged with respect to each other. So, that is important basically. So, some proteins have more than one polypeptide. More than one polypeptide chain, and the individual chains are called subunit. Are called subunits. A protein with a single subunit is called a monomer. With a single subunit is called a monomer. A monomer. One with two subunit, dimer. One with two subunits called dimer, and similarly, if three subunits, trimer, four subunits called tetramer. So, as I mentioned that in quaternary structure, some proteins have more than one polypeptide chain, and the individual polypeptide chains are called basically subunits, and basically in quaternary structure we look after that how these polypeptide chains, more than one polypeptide chains are arranged in respect to each other. So, for example, if a protein is having tetrameric tetramer, basically 4 units. So, how are they arranged? To make it more clear, I will make the structure over here. So, one monomer, another one, and as it is tetramer. So, I will draw all the 4 . So, these are the particularly the subunits can be same or different here. Here I have made like two same two sames, and to exemplify it I will give example of here hemoglobin. Hemoglobin is a tetramer. For quaternary structure, hemoglobin I am exemplifying, and it is a tetramer. Hemoglobin is a tetramer. So, here it has basically alpha chain, again alpha chain, and here beta chain, beta chain. So, the quaternary structure of a protein describes the way subunits are arranged with respect to each other. A quaternary structure of a protein describes the way different 4 subunits are arranged with each other. Here you can see that in quaternary structure basically we look after that how different polypeptides chains are arranged with respect to each other, and these polypeptide chains are called basically subunits, or if it has one subunit, then it is called monomer. If it has 2 subunit, then it is called dimer. If it has 3 subunit, then it is called trimer, and if it has 4, then tetramer. Hemoglobin is a good example of quaternary structure, where 4 subunits are arranged in a specific way in the protein, 2 alpha as well as 2 beta chains. As you can see through this example, it is a tetramer. So, basically the subunits of protein are held together by same kind of the subunits of a protein are held together by the same kinds of interaction. That hold the individual protein chains. That hold the individual protein chains in a particular 3-dimensional conformation. Three-dimensional conformation. What are those? As we discussed about the tertiary structure, they are namely hydrophobic, interaction, hydrogen bonding, and electrostatic attraction, and electrostatic interactions, basically electrostatic attractions. So, now I have completed all the 4 structures primary, secondary, tertiary and quaternary structures, and I hope I am able to convince you that how these structures are gets stabilized. In the primary structure, basically we learn about the number and type of amino acid and the disulphide bridges. In the secondary structure, we look over that what are the repetitive segments and how are they arranged, and there we talk about the alpha helix and beta pleated sheath. In the basically in the tertiary structure, all other kind of stabilizing interactions play a role. And in the quaternary structures, we look after basically how if the proteins are having more than one polypeptide chain, more than one subunits, then how these subunits are arranged in respect to each other, that forms the quaternary structure. And there we that one will be exemplified by the hemoglobin. In the case of hemoglobin, which is a tetramer, it has 2 alpha as well as 2 beta units, and how are they arranged. So, to summarize all these, I will make another schematic presentation. So, let me first draw the primary structure, and then primary structure lead to the secondary structure. So, basically in the primary structure I am making the polypeptide chain. So, this is if it is NH, and then again I am making the bond over here, and then this carbonyl bond, then again so this is the peptide chain I am drawing here. Now, this represents basically primary structure. This is primary structure, what the peptide chain is. So, type of amino acids and number of amino acids. So, this is primary structure, primary. Now, this primary structure so, this primary structure in peptide chain can get folded, and that will make the helices, or you can say so, this is forms the secondary structure. Now, here peptide chains are like kind of folded, and this leads to the secondary structure, and then it then this secondary structure can again get stabilized by different kind of bond, and that will give you tertiary structure. So, here I am presenting the this is tertiary structure. Tertiary structure, this is tertiary structure, and in basically in this tertiary structure we have these secondary structure folded. Basically you can see here this is in this structure. So, this is tertiary structure, and finally, quaternary structure. So, in quaternary structure, as I mentioned that how the different subunits are arranged with respect to each other. So, this becomes the quaternary structure. Quaternary. This is overall schematic presentation for structures. For structures. This is schematic presentation for structures. So, again I will like to summarize whatever I have discussed today. We discussed about the primary structure, we discussed about the secondary structure. There we talked about how the repetitive segment in the peptide chains are arranged, and that leads to the alpha helix or beta pleated sheet. Then we talk about the tertiary structure, and finally, we talked about the quaternary structure. I will like to stop here in today's class. We will again continue with the biomolecules lecture in the next class. Thank you very much.